My name is Harriet Ngombe, and I'm going to run you through an overview of cholera. So cholera is a diarrheal infection caused by bacteria known as Vibrio cholerae of serogroups O1 and O139. It was first discovered in 1883 and was studied by Robert Koch during a diarrhea outbreak in Egypt. So Vibrio cholerae has been known to be caused by O1 and O139, but of course there are other serogroups that are there, but these have not been known to cause disease. So among these also there are biotypes. There's classical and L2 biotype. So currently L2 is a predominant cholera pathogen during this seventh pandemic. In endemic countries, cholera accounts for approximately 3,095,000 cases and deaths. So this cholera really does cause a lot of deaths, and also there are a lot of people that are affected, and therefore it is a very important pathogen, and it's also a very important disease. And that's why it even continues to be studied from time after time. So this organism is a comma-shaped gram-negative aerobic bacillus bacteria whose size varies. It's really much bigger than the normal bacteria or the typical bacteria. Its antigenic structure consists of a flagellar H antigen and a somatic O antigen. And it's because of some of these structures that it is possible to differentiate between pathogenic and non-pathogenic strains, as well as differences between certain biotypes, serotypes, and serogroups of Vibrio cholerae. So since 1817, there has been 7 cholera pandemics, and the first 6 occurred from 1817 to 1923 and were caused by Vibrio cholerae of the classical biotype, as I mentioned. The pandemics originated in Asia and also spread to other countries as well. The 7th pandemic began in Indonesia, and then they spread to other countries and continents. And also, they have actually spread even more than the Vibrio cholerae classical. So the causative agent for the 7th pandemic has actually been known to be more transmissible. So meaning as time goes on, cholera is even becoming more virulent and also more transmissible as well. So it's very important to continue studies in this area so that really we can really get to the root of some of these as well. So cholera remains a significant public health issue in low-income countries as well. So high-income countries report few cases, mostly because they are now developed. So in October 1992, an epidemic of cholera emerged from India, and this was discovered to be a new strain, and it was named O139. It was a new serogroup, O139. Some experts regard this as an 8th pandemic, but then it's only remained within Asia. So it spread through Bangladesh, India, and neighboring countries in Asia, but not beyond. flagella to reach the intestinal mucosa and attaching via papillae, it releases cholera toxin, which disrupts the normal ionic balance, causing severe watery diarrhea and rapid dehydration. So the major virulent factors, the cholera toxin and toxin co-regulated pilus, TCP, are encoded in the bacterium genome and are acquired from the environment. So we find that when we actually find this vibrio in the environment, it won't even be toxigenic, but then it acquires its virulence from other mobile genetic elements. So cholera is transmitted via the fecal-oral route, primarily through contaminated water and food due to poor hygiene, sanitation, and inadequate water supply. So the infectious dose of bacteria required to cause clinical disease varies from the source or with the source. When ingested with food, fewer organisms are required to produce disease. So about 10 to the power 2 to 10 to the power 4 bacteria cells are required when you're getting it from food. But then when you're getting it from water, you will need more of these cells to be able to cause disease. So Vibrio cholerae is a free-living organism found in various aquatic environments such as seawater, fish, sea plankton. So as I said, in the environment, you won't really find it as toxigenic, but then it will acquire virulence factors from the environment, and then that's when it will invade the human host. So cholera has two main reservoirs, that's man and environment, mainly water. As far as literature goes, animals do not play a role in transmission of disease. So the number of people, of course, who would get infected by cholera, but the people who are at risk are mostly children and also the elderly, probably because of immunity. And there's also people with blood group O are more susceptible, maybe because of the receptors that they have, and subjects with reduced gastric acid are more susceptible because cholera will not thrive in an acidic environment. So if a person has reduced gastric acid, cholera will be able to thrive so much. So when it comes to clinical presentation, when it comes to clinical presentation, cholera is clinically identified by severe dehydration. And you also see that there's symptoms such as vomiting and watery, sudden onset of watery diarrhea, but then you see less of a fever. So the diarrhea has a fishy odor in the beginning, but become less smelly and watery over time. Just to mention that there's loss of very quickly, loss of water very quickly. And this mostly this loss of water and ions leads to death. So for the lab diagnosis, laboratory confirmation involves phenotypic methods such as culture, rapid diagnosis tests, biochemical tests, and also genotypic methods such as PCR, DNA sequencing as well. But of course, culture remains the gold standard. And cholera can be cultured on selective media such as TCBS. So TCBS will help in the isolation of Vibrio cholerae. For treatment, the primary cholera treatment focuses on rehydration through oral rehydration salts or intravenous fluids. Antibiotics are used as well to reduce the duration and volume of diarrhea. And it's very important so that in severe cases, this is used, drugs are used as well. So the drugs that have been used over the years for adults has been tetracycline, doxycycline, metronidazole, and ciprofloxacin. For children, metronidazole, erythromycin has also been used. But over the years, we have seen resistance in cotrimoxazole, which has replaced with other lines of drugs as well. So we see resistance also rising in Vibrio cholerae strains, and this calls for that when these antibiotics are being given as well, they should be guided by the local resistance pattern as well. And these drugs should also just be used if they really need to be used. So in cases where cholera can be controlled just by rehydration, it is better that only rehydration is done. So there are a number of preventive measures. One is educating people on hygiene practices, and also provision of safe and contaminated drinking water to the people. And also use of oral cholera vaccine is very important. So currently there are 3 qualified killed oral cholera vaccines at Shanko, Ducor, and Uvico. These have been shown to have an efficacy of 50% over 3 years. So they're really important. So if these are all used, they really help in preventing cholera. Thank you very much.